Hi there, thanks for coming. I'm Hao. Today, I'm going to present our recent work entitled Enriching Interpretability of Equivariant Neural Networks for Protein Structures. Let's start with introducing some backgrounds. Protein structures determine their functions. They are composed of one or several chains of amino acids, which fall into specific conformations to enable various biological functions. As shown in the figure, proteins can be defined using a multi-level structure. Most existing structure-based protein function prediction methods solely use protein 3D atomic coordinates as input and apply 3D convolutional neural networks or graph convolutional neural networks. While few attempts has been made to consider more than one structural level of proteins. Besides this, there is a need to not regard different poses of protein as different proteins when making prediction. Recently, this challenge has been addressed by equivariant neural networks, and we call ENN in short. Given a protein, whatever how this protein is rotated in the 3D space, ENNs regards it like the same protein and embed this group representation to a group of transformable vector representations. Now I'd like to move on the method part. Here is our model pipeline for protein function prediction. In the first step, we construct a protein graph based on the input PDB file. In the protein graph, each node is an amino acid, whose node feature consists of its identification and the spatial coordinates of structure center. There are multiple types of edges. The edge type are determined by its amino acid sequence and the distance between amino acid nodes. The figure shows different types of edges in different colors. In the second step, we employ an ENN and learn amino acid embedding usage. In the third step, we learn protein graph embeddings using our proposed protein graph attention pooling layer, and we give it a name, pgap. With pgap layer, we can assign node importance scores according to attention weights for edges in the protein graph after model training. These scores can be further used to identify important amino acids and important regions of proteins. In the last step, we predict protein functions or property with a multi-class classifier. Then we evaluate our method on two aspects, predicting protein functions and identifying important amino acids. First, to evaluate the performance on prediction, we use the same dataset and data splitting at Hermosilla's work in 2021. Table 1 shows experimental results for our methods together with those ENNs for 4 mainstream architectures on the task. In summary, the synergy of our proposed pgap layer and SO3 transformer achieves competitive performance with existing ENN models. In the second part of evaluation, we construct a BA horse database for this purpose. It consists of a base Barabasi-Albert graph on 60 nodes and 105-node horse structure network motifs. We train binary classifiers which consist of a 2-layer GCN and a pgap layer. As a result, pgap correctly identifies the nodes in horse motifs as important nodes. Here's the reference for the paper I mentioned in this presentation. Now that brings me to the end of my presentation. Thank you so much for your attention. If you have any questions, please do not hesitate to ask.